I'm very happy to be here speaking today, and I'd like to thank Henry and Nathan and Jerry for the kind invitation to speak. Today, I'd like to tell you about some of the work that was ongoing at Introdyne Corporation prior to our acquisition by Silence Therapeutics, particularly in the area of nanoparticle-based delivery of siRNA. But before I go into the actual nanoparticle part of my talk, I'd like to briefly discuss some of the modalities that are utilized as therapeutic agents today, beginning with small molecule therapeutics. Small molecules are the most commonly used therapeutics in the pharmaceutical industry. They are useful as both agonists and antagonists of the respective targets. Furthermore, they're effective against extracellular and intracellular targets. However, not all target classes can be modulated selectively and potently. Furthermore, lead identification and optimization are generally very slow processes, but one of the very nice characteristics of small molecule therapeutics is they're relatively easy to synthesize. The second modality, biologics, which encompass therapeutic classes such as proteins and antibodies, can also be used as antagonists or agonists of respective targets, but unlike small molecule drugs, they're only useful generally against extracellular targets. They are also highly selective and potent, but like small molecules, the lead ID and optimization are slow processes. Furthermore, unlike small molecules, biologics are somewhat more difficult to produce. An emerging class of therapeutics, interference RNA, has been worked on for the past 10 years. This class of molecules is generally useful as antagonists only. However, unlike proteins and antibodies and small molecules, interference RNA is useful against all biological targets, including those deemed previously non-druggable. Furthermore, they're highly selective and highly potent and can be generated through rapid lead ID and optimization. And finally, through existing solid phase synthesis methodologies, they are very easy to prepare. To illustrate graphically what was on the previous slide, if we imagine the blue bubble that represents the genetic space that can be influenced by small molecule drugs, and the green bubble represents the genetic space that can be influenced by biologics, and we understand that there is some overlap between where small molecules and biologics are useful, it is the blue bubble which comprises the entire genetic space that is available to be modified by interference RNA. With the advantages associated with interference RNA therapeutics, there are significant challenges. Like biologics, they must be injected because they would rapidly be metabolized in the gut. Following injection, they're subject to several challenges and barriers, which include rapid excretion, serum instability, non-specific distribution to various tissues within the body, and finally overcoming various barricades to get into target tissues. Assuming the interference RNA can get to the target tissue, there are additional challenges in achieving cellular activity. Cellular uptake of interference RNA generally occurs through endocytosis, resulting in encapsulation of the molecule inside of an endosome. In order for it to elicit its biological response, the endosome must rupture, releasing the siRNA into the cytoplasm. The siRNA can then bind to the RNA-induced silencing complex that destroys messenger RNA and results in gene silencing. In order to get around many of the problems associated with interference RNA delivery, companies have focused on solutions ranging from the simple to far more complex. In the simple and local delivery of native siRNAs has been studied, but because of the rapid lability of this class of molecules to biological processes, the tissue selection has been somewhat limited, and this has only been studied generally in ocular or lung tissues. The advantages lie in the fact that this is a very simple approach, but the disadvantages are the limited applications in ocular or lung applications, as well as innate immune responses that result from delivery of native siRNA. To get around some of this, chemical modifications have been studied. Now, such modifications to siRNAs are well known, and among the most common are methylation of the ribose ring. Such modifications are known to protect the siRNA from degradation. They can enhance siRNA potency, and they can also reduce off-target effects and cytokine induction. However, such modifications may also alter the potency of the siRNA molecules and the safety. What I have not previously mentioned is that interference RNA is a natural biological process, so these short sequences naturally occurring in the body are natural RNA oligomers. By changing them to methylated versions, for example, we are making an unnatural compound which the body will definitely not treat the way the native siRNA is treated. So in order to get around these problems, whether it's a native siRNA or a chemically modified siRNA, vectors have been studied as a means of getting siRNA to target. Some of the first vectors studied were gene delivery vectors, which include viruses, plasmids, and bacteria. Now, early work in gene therapy has utilized such vectors to deliver DNA, but relatively limited work on RNA is available. These vectors are known to induce long durations of action and be able to achieve high intracellular concentrations. However, these vectors are also known to induce nuclear transfection of their payload. This would render the siRNA ineffective because the RISC complex resides in the cytoplasm. Furthermore, these vectors are highly immunogenic, manufacturing is difficult, and there are significant regulatory and safety hurdles associated with them. This has led many companies to explore the use of synthetic vectors such as liposomes or polymers. These type of vectors, like gene delivery, are capable of protecting the siRNA or chemically modified siRNA from degradation. Furthermore, they can increase the circulation half-life, as well as increasing the siRNA cellular uptake and inducing selective tissue delivery. Disadvantages reside in the observation that such liposomes and polymers are subject to rapid accumulation in the liver. There are safety issues, and it would be advantageous if the components that make up the liposome and or the polymer are biodegradable. The final formulation should, of course, be non-immunogenic. At this point, I'd like to move into the work we had carried out at Intradyne. And to illustrate what we were working on, we had 3 platforms. One was initial siRNA sequence identification. The second was next-generation structural features, chemically modified siRNAs. And the final is the actual platform used for delivery of the siRNA payload. We had strong IP in all 3 of these categories. Regarding the siRNA sequences, effective target selection plays a critical role in the development and optimization of RNAi therapeutics. Specifically, regarding the role of the target in disease modification, in which cell type the target is expressed, and finally taking into consideration target-specific effects of specific disease models. At Intradyne, our siRNA sequences were all double blunt-ended siRNA molecules that were identified utilizing a combination of bioinformatics algorithms and proprietary know-how. The sequences were selected primarily based on extremely high potency, with IC50s ranging from 10 to 200 picomolar. Finally, our sequences were engineered to include cross-species sequence homology, for example, from mouse to monkey to man, which would give us a reasonable certainty that as we moved into disease models in mice, we would be reasonably confident that we would see a similar response as we moved up into higher animals. The next generation structural features that we had studied at Intradyne focused primarily on the Zamor design rules, known to significantly improve the potency and efficacy of interference RNA therapeutics. Such rules include the incorporation of mismatched base pairs, as well as the incorporation of double-strand overhang. These modifications are known to be effective because they increase the lability of strand separation of the antisense strand from the sense strand, allowing the antisense strand to carry out its biological response. At this point, I'd like to focus now on our actual delivery vehicle. The challenge associated with delivery of siRNAs are to create safe and effective systemic delivery technologies, enabling the delivery of siRNA therapeutics to specific tissues. Challenges also are to enable intracellular delivery as well as cellular uptake. We want to overcome inherent biological instability of the siRNA molecules and to address potential toxicities such as immunogenicity and cytokine release that are caused by the siRNAs and or the delivery vectors or the components that make up the vectors. At Introdyne, our technologies included a proprietary process for creating nanoparticles that combined a siRNA molecule with a biodegradable polypeptide. This resulted in the generation of a safe systemic delivery of siRNA sequences that was capable of not only intracellular delivery under pharmaceutically relevant conditions, but was capable of incorporating any siRNA sequence against any disease target, allowing for the simple and rapid development of new RNAi therapeutics. And all of these delivery vehicles were based upon Polytran, a proprietary family of biodegradable peptide-based polymers optimized for efficient siRNA binding and delivery. To illustrate how our system worked, Polytran, our cationic polymer, was combined with the anionic siRNA. Under conditions that we had developed at Introdyne, we were able to consistently generate nanoparticles held together by the electrostatic interactions between the two species. Our controlled conditions allowed reproducible formation of nanoparticles with diameters of approximately 100 nanometers. These naked nanoparticles were ultimately evolved into nanoparticles containing steric polymers such as polyethylene glycol, and then to introduce extracellular binding ligands. With respect to the polymer itself, we did study other polymers such as polyethylene imine and others that I don't really want to mention today. Our focus was primarily on Polytran for our first generation particles, and the focus was primarily on siRNA condensation, cell internalization, and endosomal escape. Unfortunately, these naked nanoparticles were highly labile to degradation in plasma and in solutions of high electrolyte concentrations, rendering their ability to protect the siRNA molecules far inadequate for biological use. That having been said, we did in fact form spherical nanoparticles as shown by these TEM images, and you can see that the diameters were as advertised, 100 nanometers across. Moving into our second generation, the first goal was to induce steric stabilization by incorporation of PEG on the surfaces. Doing this, we were able to extend the half-life as well as enhance tumor accumulation. After that, our goal was to put on a targeting moiety so that we could take advantage of selective binding and receptor-mediated endocytosis. I will first describe our pegylation methodology. Now, you might be asking if we took our naked nanoparticles and grafted PEG onto the surface. We did in fact have a program in that area, and I'm not going to talk about that today, but it was just published in BMCL. We found it actually more efficient to pegylate our Polytran molecules prior to formation of our nanoparticles. Polytran, illustrated here, is a custom-prepared peptide made entirely by solid-phase synthesis. It is tetrameric in nature and based on a histidine-lysine motif. It has 4 terminal lysine groups, is highly water-soluble, and as I've previously mentioned, optimized for siRNA binding. Pegylation utilized commercially available MPEG aldehyde. The method for pegylation was selective for the alpha amine. At pH 5, all of the terminal amines of the lysines were protonated, leaving the alpha amines the only available nitrogens for reductive amination. The resulting products were easily purified by ion exchange chromatography, giving a 30% isolated yield. Now, why only a 30% isolated yield? These conditions utilized a one-to-one mixture of PEG versus polytran. And as you can see, we observed a statistically relevant distribution of monopegylated material to dipegylated material and even tripegylated material, leaving measurable amounts of unfunctionalized polymer. By ion exchange chromatography, we were able to effectively remove all of the desired components and isolate only the monopegylated polymer. The molecular weight was confirmed by MALDI-TOF, giving us only the mass and m/2 peaks with no additional peaks of concern, and final confirmation was achieved by proton NMR. The top spectrum here shows the spectrum for polytran itself. The triplet highlighted represents the proton on the alpha position of the lysine. This, when integrated versus the histidine, shows exactly 4 protons. When we pegylate, not only do we see incorporation of a new PEG signal, the methoxy group associated with the MPEG is very well defined and integrates 1-to-1 to the triplet corresponding to the lysine alpha protons. Furthermore, when we integrate this triplet against the histidines, it comes out to 3. So we now have actual confirmation that the product is monopegylated. Now, as I had mentioned on a previous slide, the naked nanoparticles were highly susceptible to degradation in plasma and under high electrolytic conditions. So we incorporated a salt stability assay using 100 millimolar sodium chloride. And as you can see in the red graph, shows the instability of the naked particles. But incorporation of as little as 5% PEG on the surface of our nanoparticles significantly retarded the degradation, and incorporation of only 20% essentially removed the issue of instability for our particles. So now we had the ability to deliver far more stable particles in vivo, but how do we take advantage of the active transport associated with extracellular targeting ligands? In order to achieve that, we needed a heterobifunctional PEG. And that was achieved utilizing this PEG, which was at the time commercially available. It was a succinimidal ester vinyl sulfone, and first we would react a ligand such as RGD to displace the succinimidal ester and then use the vinyl sulfone to conjugate to our polymer. However, shortly before I joined Introdyne, this was discontinued as a commercial compound, and we were left with the challenge of developing our own novel heterobifunctional PEG that could be used for our purposes. An answer to getting this material was published in 1990 by Samuel Zalipsky, in which polyethylene glycol was chemically modified with ethyl bromoacetate. The ester was then hydrolyzed, and the resulting heterobifunctional material was isolated through ion exchange chromatography. This material could be efficiently prepared in 35% yield, and again utilizes only a single purification step. Moving forward with this product, the sequence shown here involves initial conversion of the carboxylic acid to a methyl ester, conversion of the alcohol to a mesylate with methane sulfonyl chloride, displacement of the mesylate with mercaptoethanol and sodium hydroxide, which also resulted in hydrolysis of the ester to a carboxylic acid. This was inconsequential in the following step, which was simply oxidation of the sulfide to a sulfone. The ester at this point needed to be reintroduced under the same conditions as step 1. Finally, treatment with methane sulfonyl chloride and diisopropyl ethylamine converted the alcohol to a mesylate, which spontaneously underwent beta elimination, forming the vinyl sulfone. Carboxylic acid was liberated with base hydrolysis. So in 7 steps, we were able to convert the carboxymethyl PEG to the vinyl sulfone carboxylic acid, utilizing no critical purification steps and achieving an 80% yield overall. Furthermore, this sequence was easily scalable and could be achieved in less than 2 weeks. The final step was conversion of the carboxylic acid to an active ester utilizing nitrophenyl trifluoroacetate, giving a 98% yield of the desired heterobifunctional PEG, producing a pure, a stable product, the formation of which could be monitored by thin layer chromatography and purification through a single recrystallization from isopropanol. Well, with this new heterobifunctional PEG, we did have several advantages. The first being there was only one ion exchange chromatography. The second was we had a 27% overall yield for 10 synthetic steps, utilizing only inexpensive reagents, making this a highly economical and scalable sequence. It could be completed in a short period of time, and the product was stable and storable. With the advantages, however, we found several disadvantages, specifically that we had 10 synthetic steps from PEG, that the yield was 27%, and the vinyl sulfone turned out to be problematic for our conjugation purposes. Vinyl sulfones are far more useful to conjugate to sulfides than amines. In the case of polytran, the vinyl sulfone resulted in non-specific conjugation to our polymer at any and all lysines across the entire chain of all of the tetrameric arms. This made it difficult for us to localize where the PEG was attached. Furthermore, the yield was significantly lower than what we were able to achieve with aldehydes. So this led us to develop a novel heterobifunctional PEG that would replace the vinyl sulfone with a latent aldehyde. That sequence is shown here, beginning with polyethylene glycol. Reaction with di-succinimidal carbonate and pyridine gives a 99% yield of this PEG di-succinimidal carbonate. Sequential treatment with first diethoxypropylamine and then beta-alanine ethyl ester gave us a mixture of heterobifunctional PEG and two homobifunctional PEGs. Base hydrolysis of the esters to liberate the carboxylic acids gave us the same mixture of carboxylic acid-based heterobifunctional PEGs, which could be purified by ion exchange chromatography, giving a 39% yield of the desired heterobifunctional material. Activation of the carboxylic acid with di-succinimidal carbonate gave us the final product. Now, this compound, our second generation PEG, had several advantages over the first generation. We had converted a 10-step synthesis to a 4-step synthesis. Both products required one ion exchange chromatography. While the first generation could be prepared in 27% overall yield, the second generation could be prepared in 32% overall yield. Now, while the 5% increase may not seem significant, it does represent greater than 15% increase over 27% by itself. Both sequences utilized only inexpensive reagents, making them highly scalable and economical. They could be prepared in short sequences or short periods of time. Both were stable and storable. And finally, where the first generation contained a vinyl sulfone, the second contained a protected aldehyde. Conjugation with polytran is shown on this slide. Initial treatment with RGD modified with a lysine unit for conjugation purposes gave us the RGD modified PEG. The diethylacetal could then be hydrolyzed with trifluoroacetic acid, giving us an in situ formation of the aldehyde, which was added directly into a solution of polytran at pH 5. Introduction of sodium cyanoborohydride completed the reductive amination, giving us the pegylated polytran containing a targeting ligand. As with MPEG-aldehyde, we achieved a mixture of products, mono, di, and tri products, as well as unreacted polytran. This could be purified by ion exchange chromatography to give us the desired material, which was confirmed by MALDI-TOF by molecular weight. Proton NMR allowed us to verify that we had, in fact, achieved monopegylation event. The triplet compared to the histidine signals showed 3 protons, and this was also confirmed versus the phenylalanine signals. So in summary, PEG polymer conjugation chemistry was established utilizing vinyl sulfones and aldehydes. We had demonstrated the ability to formulate siRNA with pegylated polymer. Furthermore, this resulted in improved nanoparticle stability versus naked particles. Finally, we had novel heterobifunctional pegs that were prepared in short, high-yielding synthetic sequences, providing stable, storable materials that were convenient for conjugation with both extracellular and binding ligands, as well as cationic polymers. At this point, I'd like to thank the scientists I've had the pleasure of working with. Frederick was responsible for all of the conjugation chemistry. Zhongli, Ding, and Bing Liu were responsible for formulation of our nanoparticles. Samuel Zalipsky was our Vice President of Technology Development. I'd also like to thank the other scientists I've had the pleasure of working with at Introdyne. Again, I'd like to thank the organizers of the symposium for the opportunity to speak, and I'd like to thank you for your attention.